Hey guys, it's Melissa. I just wanted to kind of quickly go through this week's presentation for you since we won't be meeting in person this week. I'm looking forward to seeing you guys next week. So let's kind of jump into this stuff. So my Zoom office hours, Wednesday, 1 to 3. You guys should have the link on your Blackboard. If you have any problems accessing that, please just email me. If that time doesn't work for you or something comes up, just email me and we can schedule something by appointment. No problem at all. Normally we do our presentations now, but we're just going to skip right through this. So I wanted to quickly go over topoisomerases. I think this is from over a week ago, probably the end of the first week or second week. So we're still thinking about double-stranded circular bacterial DNA. These enzymes cut one or both of your DNA strands and the helix either winds or unwinds before those ends are resealed. In E. coli, there's two types. Type 1 is going to reduce the number of supercoils in your circular DNA molecule, as opposed to type 2, which introduces negative supercoils into the DNA. So 1 is just going to reduce that rise, that W, if you remember from last class, while type 2 will introduce more of those supercoils. Type 2 is thought to bind to the DNA, twist, cleave both strands off, and then pass through the loop. And then once all those phosphodiester bonds are reformed, the supercoils form spontaneously, and there can be one or more than one, just depending. This slide just kind of shows that in a picture. This is your relaxed circle. If you were to use topoisomerase 2, then you can end up with a structure over here that shows those negative supercoils. So polytene chromosomes are made up from anywhere from 1,000 to 5,000 identical DNA molecules arranged side by side. It consists of both bands and puffs. Your bands are regions of very highly condensed DNA, and since it's so condensed, those regions can't undergo any transcription. And then we have our puffs, which are regions of very high gene activity, lots of transcription going on here, and there's a very high level of uncoiling of DNA. So our bands are not undergoing transcription, highly condensed DNA. Puffs, on the other hand, lots of uncoiled DNA and lots of transcriptional activity. Then we've got our lamp brush chromosomes, which exhibit extensive DNA looping. They are the site of gene activity transcription, and these lamp brush chromosomes are analogous to those chromosome puffs that we were just talking about. The only difference really between these two is where they're found. These lamp brush chromosomes are found in vertebrates, not mammals. So that's good to know, like a difference between the two. Why do we care? Why are they important to us? So both the polytene and lamp brush chromosomes just show that our chromosomal DNA exhibits varying levels of compaction and uncoiling. And those uncoiled or looped out DNA regions are active in transcription. So that kind of means that if we go back to the puff over here, this is our puff. See how the DNA is kind of looped out? It's not as condensed as in these bands. So this is where transcription is occurring, as opposed to these dark bands, which are just very condensed DNA, and those areas are not active in transcription. Histones. So histones are positively charged proteins, and this is due to the large amounts of lysine and arginine present. Both of those have positively charged side chains, which give it its positive charge. This is really important because, as we know, DNA is negatively charged because of those phosphate groups. So there's a lot of electrostatic bonding taking place here between the DNA and those histone proteins. As you can see down here, it's of course sort of like a flow chart. you have your DNA and your positively charged histone proteins. And as you can see, the DNA starts to wrap its way around there until you end up forming nucleosomes. And then you've got your chromatin structures and eventually your chromosomes. So it's kind of a good little flow chart just to kind of remember what the bigger picture is here. You discussed a bunch of modifications of your histone tails in class, and I just wanted to go over it briefly. Acetylation involves adding acetyl groups to those positively charged amino acids. So add it to that lysine, for example. It changes the net charge, and this ultimately makes it so that the histones bind less tightly to your DNA, therefore increasing the gene activity at that site. So the same kind of deal for methylation, you're adding methyl groups, and that'll help decondense your DNA, therefore increasing gene activity. Phosphorylation, once again, you're just adding your phosphate groups. Many genes can become activated, but it's good to know that genes may also be repressed depending on which sites you're targeting for the phosphorylation. So just good to keep that in mind. I included this quick little diagram about histone acetylation just to get you a good kind of image of what I'm talking about. So you start with your kind of closed, tightly bound structure up here. And then we use something like, what is that enzyme? I think it's histone acetyltransferase, I believe. HAT. So you're introducing that acetyl group, and then you wind up with a structure more loosely packed, and it has those little tails, the acetyl tails sticking out. So that's what I'm referring to. You can look this up for both methylation and phosphorylation if you're interested, but this is just kind of a good visual. So jumping into prokaryotic versus eukaryotic cells, you should all kind of know like the main difference here is that prokaryotic cells have no membrane-bound nucleus. They're found in things like bacteria and archaea, as opposed to eukaryotic cells, which have a distinct membrane-bound nucleus, and different eukaryotic cells can be found in protists, plants, fungi, animals. So we have eukaryotic cells. While these are the main differences, they do share commonalities like plasma membranes, cytoplasm, DNA, RNA, ribosomes. That kind of stuff is common to both types of cells. So keep that in mind. They do have tons of similarities. So what is the name for loosely packed DNA? That is going to be our euchromatin, and that's our uncondensed, loosely packed DNA that allows for gene expression. And then we've got our heterochromatin, which is the name for our tightly condensed DNA. That's tightly packed and it doesn't allow for gene expression. So this is kind of what we've been talking about up until this point. You see over here, the DNA is more loosely packed. So that's when we can have transcription, gene expression going on, as opposed to these more condensed heterochromatin structures, which don't allow for gene expression due to how tightly they're packed. All right, so we're going to jump into homologous chromosomes and sister chromatids, sorry. Our homologous chromosomes are pairs of chromosomes that are similar in both shape and size. They carry genes controlling the same inherited traits, but that doesn't mean that the genes are exactly the same. They can carry different versions of the same gene, and that's known as an allele. An allele is a different form of the same gene. So each locus is in the same position on the homologs, but they maybe are controlling for different versions of that inherited trait. So for example, if you get one from mom, one from dad, you can kind of have that same, let's say one's coding for blue eyes and one's coding for brown eyes, just to keep it super basic. You can have that, right? So That's what we're looking at when we are talking about homologous chromosomes. And then we've got our sister chromatids, which are identical copies formed by the replication of a chromosome. So these are identical. They contain the same allele at the same locus, and they're found on the same chromosome. So these are your sister chromatids over here. I think a pretty good way to kind of remember these is you can think of them as like identical sister chromatids, right? You know, they're kind of like identical twins, if you will. That might be kind of a good way to help you keep that straight in your head for the exam. All right. Just to quickly go over this, I know you saw this slide in your lecture, but I wanted to emphasize the diploid and haploid number here. So all of our somatic cells are present as homologous pairs, and that's diploid. We as humans normally have 46 chromosomes or 23 pairs, and that represents our diploid numbers. So we've got 46 chromosomes, that's our 2N, as opposed to haploid cells, which are our sex determining chromosomes. Those are haploid. So either our sperm or egg cells will be haploid, as opposed to our somatic cells, which are all diploid. Really important to keep that straight, especially when we talk about things like mitosis and meiosis. Cell cycle. So this is a process in which your cell grows and divides to create a copy of itself. It's composed of interphase and mitosis. What are the 5 stages? You should all be pretty comfortable naming these and understanding what's going on during each of them. So we can kind of run through this quickly. Let's get to it. Interphase. So your chromosomes are extended and uncoiled, forming chromatin. You can see here in this kind of diagram, they're kind of all over the place, just really stretched out, extended, just kind of there. And then we move into prophase, where your chromosomes coil up and they condense more. As you can see here, they're forming more condensed structures. Then we enter prometaphase, and you can see that your chromosomes are clearly double structures. It's kind of that X shape that we're familiar to seeing when we think of a chromosome. And then we enter metaphase, where those are going to line up on that metaphase plate right in the middle there. The top views here are microscopic views. So that's what you would see if you were able to look at a cell as it was going through this. That's what metaphase would look like. You can see them all kind of lined up there. Then we enter anaphase, where the daughter chromosomes start to migrate to opposite poles. So you can see they kind of left that metaphase plate and are starting to pull in either direction until you enter telophase, where those chromosomes finally arrive at those poles, and then cytokinesis can begin. So that's mitosis in a nutshell. This is meiosis, which kind of follows the same steps with the addition of synapsis and crossing over during prophase 1. Very, very important. And then the fact that they have 2 divisions, because we end up with 4 daughter cells. So just quickly, if there was no crossing over, there would be tons of issues for us. A huge one being the lack of genetic variation. That one step of crossing over creates a ton more genetic combinations or potential possibilities. And if we didn't have that, it would really reduce the amount of variation within families, within our species as a whole, and it could be detrimental. So definitely important that we follow that and understand why that's important. Other than that, like I said, one of the main differences here is that you end up with 4 haploid daughter products at the end. Definitely different from mitosis. So those are kind of like the main differences. And then I've got this little chart, I think again from your PowerPoint last week, just comparing and contrasting mitosis and meiosis. So DNA replication occurs at the same time for both processes, once during interphase before any sort of division begins. But the number of divisions is different for both of them. Mitosis goes through 1 division while meiosis goes through 2. There's no synapsis in mitosis as opposed to the synapsis occurring in meiosis. The number of daughter cells. So from the process of mitosis, you end up with 2 genetically identical diploid daughter cells. In meiosis, you end up with 4 haploid cells that are genetically different from the parent and each other. So they're 4 unique haploid daughter cells. And their role in the body for mitosis is to allow a zygote to grow or to produce cells for growth and tissue repair. So our body's kind of going through that process all the time to help repair our tissues or just help us grow, as opposed to meiosis, which produces gametes for us. It reduces its chromosome number by half and then helps us introduce genetic variability. I attached this video and I do encourage you to watch like the first minute of it. I find it really helpful to just talk through like your starting materials versus what you're ending up with here. It's kind of a good video for that. I also would encourage you to look at YouTube for some kind of diagrams and more animated visuals. I find it really helpful when explaining these processes. I like to think of your starting material versus your ending material. So you have your starting material here and you end up with, like I said, 2 identical daughter cells. And then over here, you have your starting material and you end up with 4 haploid daughter cells. So check this out. Okay, so I also think it would be wise of you to be comfortable with your cell cycle regulation checkpoints. Check out this kind of little chart here and just make sure that you're comfortable with the different checkpoints and what they're actually looking for during that process. So let's just go through these super quickly. If our human cells normally have 46 chromosomes, I'm just asking you to tell me how many are present in each of these different phases. I'm going to run through the answers, and then if you're going through this and have any questions, please just email me. During metaphase of mitosis, you will find 46 chromosomes present, and each of those has 2 chromatids, right? Like 1/2 of the X. So you'll end up with 92 chromatids. The vocabulary here is pretty important because on an exam, he could ask a question like this and maybe instead of having 46 chromosomes as a response, he'll give you 92 chromatids, and that's equally as correct. So definitely just keep that in the back of your head, the different terms that we're using during these lectures and during the questions, just so that you're very comfortable for an exam. So during metaphase 1 of meiosis, you will again have 46 chromosomes present or 92 chromatids. And during telophase of mitosis, I think this one's a little bit tricky, but yet again, you will have 46 physically separate chromosomes, but on each kind of side, so in each of those 2 about to be formed cells, those 2 cells haven't come apart yet, but as you remember, or you can flip back to what that looks like, you can see that you have the chromosomes on either end or either pole there. So they're about to be formed. There's 46 on either side. So if you've got a cell containing 40 chromatids at the beginning of meiosis, how many would it contain, or how many cells contain? It would produce, I'm so sorry. A cell containing 40 chromatids at the beginning of meiosis would, at its completion, produce cells containing how many chromatids? So we have a parent cell containing 40 chromatids. And at the end, we're trying to figure out how much genetic material is present in each of those daughter cells. So since this is meiosis, you want to be thinking what's our starting material and what are we going to end up with? So for meiosis, our final product is 4 haploid daughter cells, right? So knowing that if we have 40 chromatids that we're working with and we have 4 end products, each daughter cell will contain 10 chromatids. And again, that's kind of just you're thinking about what you're starting with and then what our final product is. And that's how you get this answer. So what condition is evaluated at the G2M checkpoint? In this case, it's going to be 1, precise replication of DNA. As I said before, I encourage you to become familiar with these checkpoints and just kind of be familiar with answering questions related to them. In the case that they are on your exam, I want you to be totally prepared. All right, that is all I have for you. Your quiz will be online. If you have any questions, please let me know. Thank you.